Welcome to this video on using pairwise alignment to minimize gaps and maximize overall homology between DNA sequences. For this example, we will optimize alignments by altering the parameters of two related sequences. Select your two sequences, click Align/Assemble, and choose Pairwise Align. Geneious offers several alignment methods. This video will cover the Geneious alignment function. Click OK, and Geneious will align the two sequences, producing a new document. You may need to turn off annotations and enable wrapping in the sequence viewer to see the whole alignment more easily. Alongside the alignment is a dot plot for the two sequences. You can see that the alignment contains very few gaps and regions of high mismatch. This is because the gap penalty and cost matrix settings favor mismatches rather than opening gaps. Also, pairwise alignments do not handle sequence inversions. To correct the alignment, start by locating the first region of mismatches in the alignment view. Turn on the consensus sequence and select Highlighting to make this task easier. In the highlighting options, select Disagreements to Consensus from the dropdown. With these options selected, the bases that match the consensus sequence appear gray, while mismatches and gaps are shaded. Mismatches may appear black at certain zoom levels. Mismatching regions should now have a striped effect, making them easy to locate. When you have located the first region of mismatches, identify the start and stop coordinates for each sequence and locate that region on the dot plot. In this example, the first mismatch region spans bases 443 to 609 in the original sequence and 511 to 660 in the mutated sequence. The dot plot shows that this region has some mismatches and some repeating sequences. Now realign these sequences with modified settings. Rather than selecting the original two sequences, select the previous alignment document and choose Align/Assemble and Pairwise Align again as a shortcut. The gap open and extension options are available under More Options. By default, Geneious sets the gap extension penalty to 3, which limits how far a gap can extend because each extension subtracts from the overall score. Eventually, a gap becomes too expensive compared with accepting a large number of mismatches. To adjust the settings so that gaps are favored over mismatches, change the default cost matrix to 93% similarity and the gap extension penalty to 0. Then click OK.
Now you will see that there are almost no disagreements between the two sequences. Uncheck Ignore Gaps to see that the aligner now uses gaps to avoid mismatches. In the dot plot, there is an inverted region, which you can see by selecting Reverse Complement in the dot plot options.
Remember, a reversed diagonal indicates a sequence inversion event. This means that one sequence matches the reverse complement of another sequence. In this example, the alignment algorithm has simply misaligned the whole section with the inverted region. Pairwise alignments always run in a single direction. For inverted regions, you need to realign the affected section. To do that, select just those parts of the sequence by clicking at the start of the region and dragging to the end. Make sure you select both sequences, then click Extract and Extract Region Alignment. You can then realign as before, but be sure to select Automatically Determine Direction before clicking OK. Geneious will reverse and complement the second sequence before aligning it. Now, when you compare the dot plot of the extracted region with the realigned version, you can see that the diagonal line is reversed. And that's how to do pairwise alignment of DNA sequences in Geneious Prime. To learn more, visit geneious.com.